All right, the next macromolecule that we're going to study is lipids. Lipids are going to vary quite a bit in structure. They tend to be but they do have a couple of common characteristics. They tend to be large, nonpolar molecules that are insoluble in water. They tend to be hydrophobic. They do not like the water. Now, this is due to their long hydrocarbon chains. Do you see this great big long chain? It's a long hydrocarbon chain. Hydrocarbon means that there is no functional group attached, or there's no non-carbon molecules, carbon or hydrogen atoms attached to this molecule. That long hydrocarbon chain is non-polar. Those hydrogens are covalently bonded to carbon, are non-polar. They tend to not form hydrogen bonds with water molecules. So those are just some of the key characteristics of lipids. Now, lipids have a couple of interesting functions, and you may be familiar with a few of these. Number one, long-term energy storage. This is typically what we think of as the purpose for lipids. However, they do have a couple of other key functions, like number two, structural components. They do form a very important structural feature for a lot of organisms. They're also responsible for heat retention, cell-to-cell communication, and protections. And we're going to walk through some of the different types of lipids and their functions. Here we have a table 3.3. I do recommend that you spend a little bit of time going through this. Not so much for anything that we are going to be memorizing necessarily, but we're going to talk quite a bit about the difference between fats and oils and where they are located, whether they are used for energy storage and insulation in animals or plants. And then we're also going to spend a lot of time with phospholipids. Phospholipids are going to be a main component of the plasma membrane, and we are going to spend a lot of time talking about the cell and the plasma membrane. So we're going to see those phospholipids quite a bit. So there are many different varieties. They all have their different purposes. Waxes, for example, serve a protective function. A lot of times they're antifungal or antibacterial, and they're also serving as water retention. So we're going to go through each one of these types of lipids. But we're going to start off by looking at the structure of lipids as a whole, specifically the triglycerides, for the purpose of long-term energy storage. As we stated previously, lipids don't really form polymers because they are made up of two different types of molecules, and they aren't really repeating subunits all joined together. They're going to be made up out of glycerol molecules and fatty acids. Triglycerides, meaning three, are made up out of three fatty acids linked to one glycerol molecule. Each one of those fatty acids is going to consist of a very long hydrocarbon chain with an even number of carbons and a carboxyl group at one end. That COOH group. This carboxyl group over here at the end is what's going to donate an OH or donate a hydroxyl group in the event that we want to link up and form a triglyceride. We're going to do this by using a dehydration synthesis. So if we were to form this molecule, we would kick off this hydroxyl group, and we would link up this fatty acid to this glycerol molecule. If we wanted to make a full triglyceride, we would have to do that dehydration reaction 3 different times to link up 1, 2, and 3 fatty acids to that glycerol molecule. So we're going to talk just a little bit about those fatty acids that are part of that triglyceride. Now, the interesting thing about fatty acids, they can be either saturated or unsaturated fatty acids. Saturated fatty acids, like this one right up here, do not have double bonds between their carbons. We all have single covalent bonds between every single carbon. Because of this, they're going to contain as many hydrogens as they could possibly hold. Thus, that saturated means they are saturated with hydrogen. Now, the interesting thing about saturated fat is that it tends to be solid at room temperature. And the reason for this is because we have really close packing. When we have these long straight chains, these long straight lines, these different fatty acids can pack nice and tight together, which allows them to form sort of a solid matrix. So they tend to be solid at room temperature. Diets that are high in animal fats have been associated with circulatory disorders because the saturated fats can accumulate in the blood vessels and clog them up over time. Unsaturated fatty acids contain double bonds. So when we have a double bond here and a double bond here, you'll see that because we have one, or a carbon can only form 4 covalent bonds. That's what we learned at the beginning of this chapter. We can form up to 4 covalent bonds. If I have one covalent, if I have one covalent bond here to this carbon, one covalent bond to this hydrogen here, and 2 covalent bonds to this carbon over here, I can't form another covalent bond. I can't put another hydrogen right here. And so we're going to be, in a sense, missing a hydrogen here, missing a hydrogen here, missing one here, missing one here. We're going to be missing a hydrogen everywhere there's a double bond. And so because of this, it's called unsaturated, meaning we're not fully saturated with hydrogens. So this is going to reduce the number of hydrogen atoms present in the chains. Now the interesting thing about these double bonds is that they actually cause the chain to kink. They cause that chain to bend a little bit. And when we have these kinks in the chain, it actually is going to prevent close packing. So unsaturated fatty acids tend to be liquid at room temperature. They're going to melt at a much lower temperature. And they tend to be liquid because we have all these kinks that prevents that tight packing. So we're going to be nice, loosey and goosey, and we tend to be liquid at room temperature. Because of this, we are going to find that nearly all animals use fat as their long-term energy storage because, think of it, we're animals. We typically have our own source of body heat. We typically try and maintain a relatively high body temperature, and we want to be solid at all times. We don't want to be just turned to complete liquid. And so we tend to store our energy as saturated fats. Plants on the other hand tend to use oils. They tend to use unsaturated fatty acids for their long-term energy storage. Plants, if you think about it, they'll freeze in the winter, and we don't want the oil in a plant to freeze. We don't want the plants to freeze solid, so they tend to freeze at much, much, much lower temperatures than saturated fatty acids do. So if we want to build our triglyceride, we're going to start off with a glycerol molecule. It's going to be a 3-carbon molecule with 3 hydroxyl groups. 1, 2, 3 hydroxyl groups. These hydroxyl groups are polar, which makes the hydroxyl group soluble in water. So we can use water to start our degradation and our dehydration synthesis. Synthesis of triglycerides is going to involve a dehydration reaction. The carboxyl groups of the 3 fatty acids are going to react with the hydrogen, or the hydrogen that's found in the hydroxyl group of the glycerol molecule, and we're going to undergo a dehydration reaction to release 3 water molecules and 1 fat molecule made up of, or 1 triglyceride made up of a glycerol molecule and 3 fatty acid tails. If we want to break that apart, conversely, we are going to need 3 water molecules in order to break this apart into its key components of a glycerol and 3 fatty acids. So the dehydration synthesis is going to release 3 water molecules, and the hydrolysis reaction is going to require 3 water molecules. Next up, we're going to talk about phospholipids and their significance in the membrane. It's a very, very important membrane. It is kind of similar to a triglyceride in the sense that it has a glycerol group and 2 fatty acid tails. However, one thing that's a little bit different is that what we have is a phosphate group attached to the glycerol head. Now, the interesting thing about this is that this phosphate head is polar. So this group over here, we have a polar head and we have non-polar tails. What do we know about polar molecules? Polar molecules like water. Non-polar molecules, what do we know about non-polar molecules? They do not like water. So we have a very confused molecule here. One side really likes the water, one side does not like the water. Now the interesting thing is this is going to be absolutely essential for forming our cell membrane. Our cell membrane is actually not a single layer of phospholipids. It is in fact a double layer where we have phospholipids facing up and phospholipids facing down. We have phospholipids with the polar heads facing the inside of the cell and phospholipids with the polar head facing the outside of the cell. And that's because the fluid inside of your cell is mostly water. Your cytoplasm is going to be mostly water. The interstitial fluid in between your cells is also going to be composed of mostly water. And so we're going to have this sort of phospholipid bilayer where we've got our polar heads on the outside of the cell and on the inside of the cell with our nonpolar tails found in between. Now, the interesting thing about this, or one other thing I would like to point out about these fatty acids tails, is that one of these fatty acid tails is fully saturated. One of these tails is actually going to be non-saturated. It's going to have a single double bond in there, which is going to create a kink, a slight kink. Now, that slight kink allows those polar tails, those non-polar tails, to be pretty flexible. It allows the cell membrane to be flexible. It allows us to maintain a lot of movement and fluidity with that cell membrane. Next up, I want to talk about steroids. Steroids are another very, very important lipid that kind of have a very different structure than what we've seen so far. Our other lipids have been composed of a glycerol molecule and fatty acid tails. Our steroids are also lipids, but they are going to be very different in structure. They are going to be composed of 4 rings that have been fused together with various functional groups attached to that original carbon skeleton. An interesting thing that we have here is we have cholesterol, testosterone, and estrogen. Testosterone and estrogen are sex hormones that produce widely different results. An individual exposed to huge amounts of testosterone, as in the case of a male, is going to end up with very different characteristics than an individual who's exposed to a lot of estrogen, like a female. Very, very, very different end result. But the interesting thing is that the difference between testosterone and estrogen and the way it reacts in the body and its ultimate end result is all going to come down to the functional groups. It's all going to come down to the fact that testosterone has a double bond oxygen over here, is a ketone with a methane group off to the side. Estrogen does not. Estrogen has a hydroxyl group and no methane group right here, and that's it. That's the only difference. Those 2 different functional groups make all the difference between testosterone and estrogen, and both of them are formed from cholesterol. Cholesterol forms is our precursor molecule that makes all of the other steroids. So cholesterol is a very, very important part of your diet. It's an important part of the body, and we're also going to find out a little bit later that cholesterol plays a very important role in the cell membrane. The last thing that we're going to study is waxes, or the last lipid that we're going to study is waxes. Waxes are going to be long chains of fatty acids connected to carbon chains containing alcohol functional groups. So the interesting thing is about this with these really, really long chains, the alcohol groups may add some sort of polarity, but for the most part we have these very, very long non-polar chains. This means that they tend to be solid at room temperature, they have a very, very high melting point, and they tend to be resistant to degradation by hydrolysis because they're so hydrophobic. They don't, with all those long non-polar chains, they don't like water, they are afraid of water, which means that they're very difficult to break down. It requires quite a bit of effort to break down a wax by just using water, which you may have noticed if you've ever tried to wash a waxy fruit. If you've ever, like grapes in particular, you can see this nice waxy coating on the outside of a grape. You may notice that if you wash that with water, the water just sort of rolls off. It beads up and it rolls off. Pay attention next time you wash grapes, or pay attention the next time you wash an apple. The water beads up and rolls off. But if you polish it, you can easily remove that wax with actual physical force and just sort of polish polish those grapes to get that waxy coating off. I don't know if anybody else does this, but I was taught to always polish my apples. You don't just wash them, you polish them, because the pesticides and bacteria can get stuck and coated in that. There's a waxy surface that protects the apple from the pesticides. It protects the apple from any sort of insect, bacteria, or debris, but just washing it with water isn't going to get that off. You need to actually polish that wax off to remove everything on the surface. So its primary function is going to be for protection. It's going to prevent water loss from exposed regions. May also be involved in repelling insects. Earwax contains cerumen, which repels the insects and traps dirt, dust, and debris to protect your eardrum. So has a protective function inside the body.